Okay, thank you very much. I will tell you some stories related to telomeres that we are doing in the lab. And I apologize because I'm a wet biologist, so I'm not going to give a lot of algorithms. And I hope that what I say is clear enough, and if not, just ask me. So telomeres are the ends of the linear chromosomes, and they are made up of proteins and DNA. The DNA are simple repeats, and the proteins are there to protect the ends and give them a special shape in order to ensure that the telomeres can carry out their two main functions. Okay? So one of the main functions is the following. Each time a cell divides, duplicates its DNA, we are losing a little bit of information from the end of the chromosome. And this is it's called, it's a fact, okay? And after a certain number of cell divisions, the cells senesce and die. This is called the Hayflick limit, and actually gives a limit to the number of cell divisions that the cell can undergo, a somatic cell, in our body. So this sounds very sad, but actually it's a very important anti-cancer mechanism, because this does not allow the cells to grow indefinitely. You have to do a certain number of cell divisions, and then you stop. However, there are situations, for example, in embryos, in which you have to do a lot of divisions. Okay, if you think about the sperm and the egg, you make a zygote, and now you have to make a baby. Okay, so it's a lot of divisions, and so in those situations, you need some way of elongating the telomeres, and there is a special enzyme called telomerase, which is really, really special. It's a reverse transcriptase. That means it's an enzyme that takes RNA and makes it into DNA, and it uses its own template to do that. It works something like this. If you have the end of a chromosome, okay, you see the repeats, telomerase will come and use its own green RNA as a template to synthesize one repeat, and then move aside, synthesize another repeat, move aside, synthesize another repeat, and so on and so forth. And eventually, you have to synthesize the other strand of the DNA, and this is done by the regular machinery that synthesizes DNA. So we know that telomerase is active in embryos. It turns out that if you are a cancer cell and you want to live forever, the first thing that you have to do is to make sure that you have enough telomeres to be able to live forever. And in about 80% of the cases, as we heard yesterday, there is a resuscitation, if you want, of telomerase. So telomerase is usually repressed at birth. So in our somatic cells, we don't have telomerase. But in cancer cells, in about 80% of the cases, we get reactivation of telomerase. And in about 20% of the cases, there is an alternative mechanism based on recombination that maintains telomeres. The second important role for telomeres is to make sure that the cell does not repair them as if they were a broken chromosome. If you think about the broken chromosome and you think about the telomere, they look exactly the same. There is no difference at the DNA level. However, if you would try to repair something that doesn't need repair, you will actually make a mess out of the cells. Okay? So you will start fusing chromosomes, chromatids, and you would get a lot of genomic instability, which eventually will bring to more cancer. This is a very good mechanism that cancer cells use to actually generate more variability. What is quite striking is that, and this is relative to the Goa notation and the things that you can find in the literature, that many of the enzymes that are required to repair a double-strand break are the same enzymes that at telomeres are required to prevent the repair of the double-strand break. Okay? So whenever you get some kind of Go term, it doesn't really mean that you understand what the Go term does or how it is related to a certain disease. I mean, the logic is not automatic, let's call it. Okay, so we work with yeast, which gives us all the good things in life, as you know. This is the same yeast that gives bread and beer and croissant. And, but most importantly for this talk, yeast cells have expressed telomerase, and so they are like embryonic cells or immortal cells, cancer cells. And we would like to we are asking really, really basic questions. And one of the great things of yeast is that you can really use it to ask basic questions that it's very hard to answer in other systems. So the basic question is, how is telomere length regulated? We know that there are mechanisms such as telomerase that elongates the telomeres, and there are other nucleases, for example, that will shorten telomeres. And we know that each one of them is regulated by other regulators, and there are all kinds of mechanisms that keep the homeostasis of the length. So, for example, we already know, and again, I apologize, I'm not going to enter into all the details, but there is a bunch of proteins like RAP1 that binds telomeres and actually measures the length of the telomere. And there are some proteins that bind to the binding protein and repress or help recruiting the telomerase. So if the telomeres are very long, you have a repression and you don't recruit telomerase, and with the cell divisions, you lose the length of the telomeres. If you go too short, now you can recruit more telomerase and elongate. And so there is an homeostasis. So some time ago, we took advantage of the fact that in yeast we have a collection of all the possible mutants. Okay, so we have 6,000 genes, and we have 6,000 strains, each one with a mutation in a different gene. And so we screened all these mutants for all the mutants that have an effect on telomere length. And we were looking for either short telomeres or long telomeres. And we call these mutants TLM, for telomere length maintenance. And you can see here that it's easy to recognize the last fragment. So this is a southern blot, but it doesn't really matter. We are measuring the telomeres, and since we are measuring telomeres in a population of cells, each one with many, many telomeres, we get a distribution, but it's very easy to see that the wild type has always the same size of telomere length, and you can find mutants that have shorter telomeres and those that have longer telomeres. So when we did this for several collections, we have the collection of the non-essential genes, collections of essential genes. In that case, we need some genetic trick because we cannot make a deletion of the gene. And we finished having about 400 genes that when you mutate each one of them individually, you get either long telomeres or short telomeres. Now, I would like to make sure that you understand, it's not that we are saying that there are 400 proteins sitting at the end of each telomere. What I'm saying is that there are genes in the genome that when you mutate them, they affect telomere length. It could be direct, it could be indirect, there are many, many possibilities. So when you look at the list of the genes, 400 genes is a lot of genes, and you have essentially anything that you were expecting to, things that have to do with DNA metabolism, chromatin configuration, protein modifications, RNA, et cetera, but also a lot of surprises. On one hand, we get many, many genes for which we know the function, but we have no idea how this function affects telomere length. For example, vacuolar transport is one of the most enriched groups in our set, but we don't know why it affects telomere length. And we get many, many genes for which either we don't know what they do or we think we know what they do, but it makes no sense that they affect telomere length. Okay? So what do we know? What do we do if you have 400 genes that you are interested in? So you can do one of two. What I call the vertical approach, which is you take genes that you are interested in, usually P53, and then so you throw away all the list of 400 and you concentrate on one or two, and you analyze what they do, where do they act, how do they act, how they are regulated, and so on. What I call the vertical view, but it used to be called molecular genetics or molecular biology. Or you can do the horizontal view, which is to get a systems view to see how the information flows within the system, how many pathways you can recognize, how are the genes organized, and so on. So we're trying to do both. So for the system-level approach, you can, we are working with yeast. There is a lot of information about yeast. There are genome-wide analysis of genetic interactions, genome-wide analysis of protein interactions. You name it. And so we can use that vast amount of data, and if you take, for example, all the 400 genes that are involved in telomere length maintenance, and you use that data, you get a very clear picture of how telomere length is being regulated. So obviously, it's not a simple thing, okay? So we are trying in several different ways to try to organize these 400 genes into pathways or into regulatory units. And so today I'm going to tell you some of the things that we are doing. The work I'm going to talk today was initiated by Lior. Lior is an MD PhD student that finished already, and you see that it's a very serious neurosurgeon. Actually, today he's a neurosurgeon, and together with Linda and Vera. And the basic idea here was to do an epistasis analysis. Okay, so this is how geneticists have worked for years, but of course with 1 gene or 2 genes or 3 genes. You make a double mutant, and you compare the double mutant with individual single mutants, and you say the following: if the double mutant is worse off than the single mutants, that means that there is an additive effect, and they are probably working in separate pathways. And you can actually formalize this in a mathematical way. So if there are 2 different pathways, you can say, okay, if I make the double mutant and I measure, for example, telomere length, I can expect that the double mutant will have a stronger effect than each one of the single mutants. And if they are in the same pathway, the moment I already knocked out one of the 2 mutants, then I inactivated the pathway, so the second mutation should not have any effect, and I will have a number which is larger from what I would expect from independence. And of course, there are several situations in which there is synergy. For example, when there's competition or overlapping pathways, and so in that case you may have a surprise, and when you do the double mutant you get a phenotype that is much, much stronger, even lethal, than what you would expect for a single mutant. Okay? And usually this approach is carried out for fitness. Okay? So you can do it with different organisms. In yeast, usually you measure the size of the colony, and you use it for genetic interactions. We apply this for actually the telomere length itself. Okay? Yes?
Does data show a multiplicative effect like this, or could it also be additive?
Okay, so we did it both ways because it's a theoretical thing, so we can actually compare. So what we do is actually we compare the observed versus the expected, and we can expect to be multiplicative, or we can expect it to be additive. Okay, and in both cases the results are very, very similar. We couldn't distinguish between the 2 models. So essentially what we would really like to do is to take the 400 genes, 400 mutants, and do the matrix of 400 by 400 double mutants, right? So this is only 160,000 double mutants. So I said to Lior, okay, go ahead. Just take the 160,000 double mutants, extract DNA, make triplicate DNA preparations, thousands, and he looked at me. Well, we are from the Middle East. He looked at me and he said, I'm willing to do 2. And I said, okay, well, you don't have to do the 160,000. You can do only half of them because it's symmetrical. You can do only 80,000 in triplicates, and we are done. 3. And in that very systematic and highly scientific way, we arrived to the conclusion that we want to do a matrix of about 40 by 40. Okay, and at the beginning we were very naive, and we were doing actually long mutants together with short mutants. So we were crossing everybody by everybody. And then we discovered that we couldn't make sense out of the results, because in many cases you cross, for example, a long mutant with a short mutant, and you get a wild type length. And you don't really know whether this is some genetic result, double suppression, or this is a mathematical result. One adds something, the other one takes out something, So we decided to go for the long mutants separately and for the short mutants separately. So, for example, 40 by 40 long mutants is 1,600, and you won't believe it, but he did it in triplicates. So we ran a southern blot with markers for size, and we can run the triplicates of the double mutants and the single mutant that we recreate each time. And Lior wrote a program called TELQuant, which automatically actually detects the size of the bands and transfers it to an Excel sheet. And so we can actually make the calculations. And I'm not going to enter into the biology here, but the idea is that now we can actually use the epsilon terms that we derive from the results to try to put together genes that seem to be acting in the same way in the same categories. So this is what we see here. So, for example, you can notice here that there are 3 genes here that have essentially the same type of interaction. So, sorry. Yellow in this scheme means epistatic interaction. That means they tend to work together. And blue means that they work separately, okay, synergistic. And so you can see that these three mutants seem to have essentially the same kind of interaction with all the other mutants in the collection. And so what we can do from here is look for stories, for vertical stories, that we can go to the lab and do the biochemistry and really see whether they work together, they interact physically, they are at the same place, et cetera, et cetera. And you can see that there are several of these stories here. Or we can go one step back and look at the system and say, okay, we know of about 4 different ways, or maybe 6 if you separate this one in 2 or this one in 2, ways, different ways in which telomere length can be regulated or can be, these genes are in charge of preventing elongation by telomerase. Okay, so this is one of the efforts, and the other thing that we can do also is look for these magic squares. So when you find a magic square like this, in which you have a series of genes that have epistatic relations among themselves, okay, so everybody with everybody is yellow, that probably means that we are talking about some kind of complex or pathway, okay? So we are very interested in understanding who are these genes and what are they doing and how they're doing it and so on. Okay, so that was one story. The other story is what we call TELKA, telomere length kinetics analysis, Assay, sorry. And this, the idea behind this assay to try to, again, we are trying to take the 400 units and instead of talking about 400 different entities, try to say, okay, we can talk about 10 different pathways, or 5 different pathways, or 3 different pathways, or whatever. And so here the idea was to see, the basic idea is that if there are genes that work together in a certain pathway, you expect that the kinetics of shortening or the kinetics of elongating should be similar in the mutants of the same pathway. Okay, and so what we did was to create strains that are heterozygous for the mutation. Okay, so they have one copy that is wild type, one copy that is mutated. Because all these mutations are recessive, the diploid that has both copies actually has normal telomeres. And then we put these strains to undergo meiosis, and we get descendants that are either mutant or wild type. And the mutants, they start at the beginning. Once meiosis is finished, they get a set of chromosomes that have normal length telomeres, but they are mutant. So they will start to shorten or they will start to elongate according to what they should be showing, right? And so we can follow them through the generations and try to see what is the kinetics of this event. And what we found out is that if you have mutants that affect the same pathway, you get very similar kinetics. And so we use this trick to try to see if we can put together mutants into categories according to the kinetics of shortening. And I won't go into all the details, but there was one group that was group number 3, which was very small, and it was very striking. And the striking thing here was that at the first opportunity in which we could measure telomere lengths in this group, the cells had already attained the final size. Okay, so we take cells, we take diploid cells, we put them through meiosis, and then we take one spore, and we just grow enough in order to get DNA, and when we get the DNA, the telomeres are already at the final length. Okay, so that means that the shortening is extremely short, extremely fast. And when we looked at who were the genes in this category, we got the Ku couple, RNR1 and MET7. We know that the Ku couple is a complex that is very important for promoting DNA repair in broken chromosomes and preventing DNA repair in telomeres. And we got RNR1 and MET7, and MET7 was a gene that nobody knew what it was doing. We have no idea what they were doing, and so we did biochemical experiments to try to find out what they did. I'm not going to enter into all the biology because it will be boring for you, but suffice to say that we found out who these proteins were and how they were related to each other and what was the mechanism that was being activated So they work together. We can demonstrate that they can be both immunoprecipitated together. And one of them has a role in DNA repair. So the MET7, the new gene that we didn't know anything about, thanks to this methodology, we could associate it with Ku and give it a role also in DNA repair. Okay, so finally I would like to talk about nature and nurture. And there are very few things that we are certain about in life. One is taxes, the other one is death. And the third one, apparently, is that telomeres is what makes you old. Right? I mean, everybody knows that. I can read it in Cosmopolitan, and every time I talk to somebody and I said, yeah, I work on telomeres, they said, okay, so this is what makes me old, right? So it's a very well-known fact that telomeres get short with age. Okay? Everybody knows that. And actually what nobody knows is that the data behind that statement, which is the basis of companies, I mean, I used to get this letter saying, enlarge your penis. Now it's enlarge your telomeres, right? And it's probably the same pill. So there are companies that will tell you when are you going to die according to the measure of your telomeres. Okay, so this is an important stuff. So the basis for that is this paper here in 1998, which measured 12 individuals that were newborns, 24 that were their parents, 35 that were grandparents, and 4 that were great-grandparents, and found that there is a correlation between telomere length and age. Okay, so the correlation is there. However, things are not so simple, and the more we know, the less it appears to be that simple paradigm. And so, for example, now there is a paper about 100,000 subjects to which they measured telomeres, and you can see here that for women there is a completely different curve for women. Okay, women, their telomeres go down with age, and by the age of about 30, they stop shortening, whereas men continue to shorten their telomeres until the age of about 70, and here probably there is some kind of selection. But I think that most interesting is this paper here that actually followed a bunch of 10,000 people in the Netherlands, and then they measured the telomeres with 10 years apart. Okay, so this is the first horizontal study in which they took one person and measured its telomeres, his or her telomeres, 10 years after, okay? And this is the result, okay? You can see that about half of the people, the telomeres got elongated, half of the people, the telomeres got shortened, okay? Moreover, I think that this is what we should be showing everybody, okay? This is the famous correlation now measured in 10,000 people. So what you can see is that, indeed, there is a correlation between age and size. The older you get, the lower the size of the telomeres, right? But there are young people that have shorter telomere than old people. So just blindly taking a person's telomere length and using it to decide when he or she is going to die is impossible from this data. Yes?
How consistent is this across the cells?
Okay, so all the experiments that we are showing were done with white blood cells. Okay, so we were trying to do it with brain, but we couldn't get volunteers to give samples. So it's not being done. There is no data whatsoever from any other tissue.
Yeah, so I had a very related question. I mean, so those cells probably were created from stem cells not so long ago.
Okay, so
different for things that don't have.
Okay, so I'm just showing, the things that have been published, and you can take home messages. But what started our analysis was actually this paper, which really, really struck me, because the title of this paper was Accelerated Telomere Shortening in Response to Psychological Stress. The basic idea was they took a bunch of people very, very stressed and a bunch of people very, very non-stressed, and they compared the telomeres length, and they decided that psychological stress shortened your telomeres. Okay, So the teleology behind this is quite simple, because if telomeres really get shortened with age, and people that are stressed look older, right? White hair, and you don't sleep, and so on. And so makes sense, right? And for the graduate students, the very, very stressed people were not graduate students, okay? And the non-stressed were not PIs. But still, I mean, I said this is very, very weird, okay, to do. Okay, so we decided to use yeast cells because we can do with them what we want. And so we said, okay, let's take all the different ways that we can imagine of stressing yeast cells, not psychologically, because I have no idea how to do that, but we can actually stress the cells in the sense that we can make them grow, I mean, use a stress that makes them grow slowly. And so we used a series of all the possible stresses that we could think about. And when we applied that to yeast cells, okay, so this is work initiated by Gal and Tal, and continued by Yaniv. This is Yaniv, not this. And Vera. And what we found out is that most of the stresses don't do absolutely anything. But there are some stresses that do have an effect. For example, caffeine will shorten your telomeres, and ethanol will lengthen your telomeres. Okay? So now you know how to balance your life, right? So this is the data. Actually, what you can see here is that it's not only ethanol. Most alcohols will elongate the telomeres, but ethanol is the one that works better. Okay? You can see here, every time you see the arrow, that's the wild type. And you can see that ethanol, methanol, isopropanol, ethanol, all of them elongate the telomeres. Acetic acid will do something similar. Caffeine will shorten your telomeres and also high temperature. And hydrogen peroxide, which is oxidative damage, which was the interpretation of the psychological stress. So what they said is that psychological stress causes oxidative stress in the cells, and that is what is shortening your telomeres. Didn't do anything. Okay, so at least for yeast, we don't see any effect of oxidative stress. And if you add caffeine, what you see is that with the generations, you see a shortening of the telomeres, which does not get to the lower possible length, but it gets to a plateau. And if you wash the caffeine away, the cells return slowly to the normal size. So this is not a mutation that you are selecting for. This is a physiological response to the presence of caffeine or ethanol. So with ethanol, the image is very, very different. What you can see here is that the cells start with short telomeres, normal telomeres, and then as long as you keep adding ethanol to the culture, the telomeres get longer and longer, but also you can see that the variability is larger and larger, which means that the mechanism that chooses the short telomeres to be the ones that get elongated is lost, and you actually now elongate everybody. And so you get this smear, yes.
So you're talking about, like, not the molecular mass. You think the molecular mass is staying the same, but the physical length is shrinking. Is that?
Yes, I'm measuring the telomere length of each culture. Yes. So telomeres kept growing. We have now strains that have 10 and 20-fold longer telomeres than the normal wild type, without any effect that we can measure. So we can do competition experiments between the long telomeres and the normal telomeres. We don't see any difference. Okay, so this is actually a very great situation to be in, because we have now a list, of all the possible genes that affect a certain trait. And also we have environmental signals that affect the same trait. So we can actually try to see how the environment and the genes interact. So the famous nature and nurture, or environment and genes, we can actually start probing how it works. So what we did was to measure the expression of all the genes under the different conditions. And then the basic idea is that if the environment is sending some signal, and the cells are responding to that signal by elongating or shortening the telomeres, the TLM genes should be in the middle, right? Because these are the genes that cause elongation or shortening of telomeres. So we can ask, if we make a mutation in one TLM gene, will it interrupt the signal, let's say, if I grow all my collection of TLM mutants in the presence of ethanol, will I see mutants that don't elongate anymore their telomeres? Okay? And when we did that, what we found out is that in general, so what we are seeing here is what is the initial length of the various strains and what is the elongation after 100 generations, or what is the shortening after 100 generations. And you can see that the genes actually elongate, or the mutants elongate in response to ethanol or shorten in response to caffeine in a way that is proportional to the phenotype that they have without the stress. That means that each one is contributing whatever it can, right? And so we are looking for things that are outside of this range, and the clear winner is RIF1. So RIF1 is a mutant that has very long telomeres, but does not respond to ethanol, almost nothing, almost does not elongate, or does not shorten with caffeine. So to make a very long story short, and again, I don't want to bore you with all the biochemistry, what we found is that ethanol causes a reduction in RAP1. RAP1 is the enzyme, the protein that binds the telomeres, actually serves as a ruler to measure the length of the telomeres. And because of that, we have less RIF1 and RIF2 in the telomeres. And if you replace the promoter of RAP1 by a promoter that does not respond anymore to ethanol, okay, you can see here that ethanol does not change it, then instead of this response, you get now this response. So actually, it's very important. The cells are responding to the presence of ethanol by actually reducing the amount of RAP1. So this is our model. Okay, so in normal cells, you have a certain length, which is kept. If you have a very short telomere, now the cells are trying, they count how many RAP proteins they have, and according to the number of proteins they have, they recruit more and more telomerase. And in the case that in the presence of ethanol, they have very few RAP1, but they don't know it, and so they keep, so the telomeres are long, but they keep bringing telomerase and elongating more and more. And in the last minute I have, so please join me in a prayer. In the last minute I have, I would like to tell you about something very, very cool that we are doing now, these days, here in Berkeley. It has to do with heterochromatin, chromatin, and so it may interest you. One of the problems that we found is that we don't actually have a very good way a functional way of establishing, of reading heterochromatic situations. So we can actually do all these genome-wide experiments, nucleosomes, histones, things like that, and see the modifications, but we don't have actually a very good readout. And the laboratory of Jasper Ryan has developed a very, very cool thing that I just wanted to tell you in 1 minute so that you know. It's called the crash assay. And it's based on the following principle. In the yeast cell, we have silencing, that means heterochromatic regions in the chromosome, the telomeres, and also two silent cassettes, one that is called HML and the other one that is called HMR. And this is mediated by the acetylation of histone 4 at the K16. And the SIR2 complex, the sirtuins, are actually the ones that are in charge of establishing that silencing. And so what they did was to replace one of the genes in the silent cassette by a Cre recombinase. So the Cre recombinase knows how to recognize these LOXP cassettes, and they inserted somewhere in the genome an RFP gene bracketed by the two LOXP cassettes. So if this gene is silenced, there is no Cre recombinase, and the cells are red. However, if the cells are red, the Cre becomes even transiently expressed, so if the repression on the chromatin is lifted even for a minute, we will get expression of Cre recombination that will turn the cells from red to green. And this is how it looks. So if you have a wild-type cell, it's mainly red, and you have very few sectors of green. So these sectors are actually because the first cell here underwent the switch, and then all its descendants actually were already switched. Okay? And we can now take these cells and use use mutants. Okay, essentially we can use all the yeast mutants to try to see how they affect the heterochromatic state. And you can see, for example, that this, my favorite mutant, ELG1, has a huge amount of this switching, whereas there are others that have a very, very low amount of switching. Okay, and this is what I wanted to tell you. And so finally, I would like to thank the people in the lab, and this was a collaboration with people from the School of Computer Sciences in Tel-Aviv University, Ron Shamir, Roded Sharan, and Eitan Rupin. And thank you for your attention. And
questions?
So all the lengthening and shortening in response to those stresses in yeast, I totally understand. I'm trying to relate it back to the human context. I thought you were saying that we don't have active telomerase in somatic cells. So no amount of drinking alcohol is going to lengthen those telomeres?
okay. First of all, I would like to tell you that we try to put alcohol and caffeine into our human cells, and they die. Okay? So it's not a simple thing, because, I mean, we are using amounts that are absolutely irrelevant to the human, behavior.
It's a little bit of a joke, but I'm just trying to understand for sure that...
So essentially,
when people talk about human telomere length, they're talking about regulation of how fast they get short, not so much...
So most people don't talk about anything significant. They actually are talking about what they measure, which is a PCR reaction that is also problematic on white blood cells, which we don't know when were they created, because they are renewed all the time. It's part of our blood, okay? So it's very hard to know exactly what that means, okay? But there is a correlation, which we don't really know the meaning of, between age and size that can be measured in that way, okay? Most of our cells express very, very, very low levels of telomerase, and it's only the embryonic, or the stem cells, the ones that are actually expressing telomerase, and so they are able to cope with whatever change is needed.
Okay, maybe we continue discussing outside and then
Okay, great.
thank Martin again.